Well, good afternoon. My name is Stephen Benner, and I am going to talk at a socially correct distance about synthesizing life, which has many attempts at a definition, but we refer to life as a self-sustaining chemical system that is capable of Darwinian evolution. And we do this because this is not only a definition of life, it's also a theory of life. And what we mean by that is that we think that the only way that matter can spontaneously assemble and deliver properties that we value in life is by a process called molecular Darwinism, which means that you have an informational system which can replicate. That replications occurs with imperfections, but those imperfections are themselves replicable. And according to this definition theory of life, natural selection can do the rest. And the only real question is, how far can that life go, and in what period of time? So why create artificial Darwinism? Well, there are two general questions. One is for science and one is for technology. Science is what we do when we try to understand something. And you have to remember that if life is hard to define, understanding is even harder. One of my favorite quotes is from Richard Feynman, who points out that people are easy to fool, and the easiest person to fool is yourself. So very often, when you think that you understand something, in fact, what you have is the illusion of understanding. And there's a way, there are several ways, but there's one way in particular that you can manage this propensity to self-delusion is through what we call grand challenge synthesis. That is, you set a big goal beyond or just beyond what you think your theory is able to allow you to achieve. Pursuing that grand challenge drags the scientists across uncharted territory, and it forces them to solve unscripted problems. Thus, if a theory is inadequate, the resulting failure cannot be ignored, the synthesis fails, the grand challenge is not met. And for this reason, pursuing grand challenge synthesis drives discovery and paradigm changes in ways that hypothesis-directed research cannot. So this is a direct criticism of hypothesis-directed research. When you craft a hypothesis, you generally make sure that the hypothesis is very likely to be true. And if you do not do that, the funding agency will do so. But in point of fact, you very rarely learn much new with hypothesis-direct research that you didn't already know. And synthesis, pursuing this grand challenge, is one of the ways in which you relieve yourself of that constraint. Of course, we can also create artificial Darwinism to do stuff. That's the technological goal. And because actually natural Darwinism delivers great stuff. But, you know, a three biopolymer system, which is what you and I do and most of the world that we know about does, with DNA, RNA, and proteins are all working together, it's actually very difficult to evolve in the laboratory. And this is why Francis Arnold and Greg Winter and George Smith got a Nobel Prize a couple years ago for doing this. It's very, very difficult with this three biopolymer system to get phenotype in proteins out of DNA, because 100% of your problems is how do you tie the protein of the sequence of the protein that is performing what you're selecting for with the DNA molecule that encodes it. So DNA holds the information in our three biopolymer systems. Proteins have functional groups, but we really want a biopolymer that has both so that we can evolve it directly to do great things. Now, actually, it turns out for models of how natural history got going on Earth, there is actually something called the RNA world model for early life on Earth. That model holds that actually RNA was just that biopolymer 4.2 billion years ago. It did both genetics and phenotype. It could catalyze reactions. It could store information. And so this is associated with what we'll call the standard model for the origin of life. In the beginning, Earth cooled, organic material formed, and you can see a scene from Star Trek with Jean-Luc Picard and Q watching the first events, which is an RNA molecule assembling by accident, spontaneously, that was able to catalyze the template-directed synthesis of RNA. And the argument is, the model is that Darwinian evolution began in this RNA world. One biopolymer RNA did both genotype and phenotype. There are two papers at the lower left-hand corner of this slide, which you can go back and look at. if you want to see how this worked and how life actually emerged in this model. But one of the things that you know about from your studies of biochemistry is that RNA is everywhere in modern life. It survived not only as an intermediary between DNA and proteins as a messenger, but RNA is actually found on cofactors. This is the magenta structures on the right-hand side of the screen, which are all pieces of RNA tacked onto functional molecules with particular chemical reactivity that are performing roles in metabolism, roles that they ostensibly performed in the RNA world left over. But there's a problem. RNA and DNA do phenotype actually quite poorly. There are few building blocks, there are few functional groups, and that's especially true if we compare those structures of the RNA or DNA building blocks in the upper right-hand corner with all the 20 amino acid protein building blocks, which have positive charges, negative charges, hydrophobic groups, hydrophilic groups, functional groups. There's clearly a lot more that proteins can do just because they have more building blocks and more functionality. But of course, that means it's bad technology, but we can actually now set as our grand challenge trying to make a better DNA or RNA by, for example, increasing the number of building blocks in the system. And we can do that by using all hydrogen bonding patterns that are available to us. And I just represented hydrogen bond donors in this Lego structure as prongs, and the holes into which they go, those are the red dots. And it's quite clear that I can increase the number of base pairs by increasing the use of prongs and holes, hydrogen bond donors and hydrogen bond acceptors, to make at least 12 letters in the genetic alphabet. That's not as many as the 20 in proteins, but we can work with that. There is the same structures now, all with the atoms drawn out. The hydrogen bond acceptors are oxygen atoms and nitrogen atoms in red. The hydrogen bond donors, the prongs, are these blue hydrogen atoms attached to heteroatoms. This is something that has been called an artificially expanded genetic information system, which is nice because it has the tidy acronym AEGIS. Now, we could also imagine putting side chains on these for a biopolymer that is both evolvable and catalytic. And actually, you can see how long we've been pursuing this grand challenge. This is a notebook from my lab. Actually, it's my notebook from 35 years ago, signed by various people who were signing it because we thought it would be patentable. We were going to now make all of these extra letters in the genetic alphabet, add functional groups just like proteins onto the side of them. And by adding functional groups to nucleic acids, we will enhance its catalytic potential, like Tom Cech's RNA, which of course had just emerged, but better. A grand challenge indeed. Well, there's been a wealth of discovery in that grand challenge as we pursued it. I'm not, of course, going to have any chance to describe to you all of the things that we have learned by asking these what if or why not questions as we've tried to increase the number of building blocks in DNA. By the way, you can get all of these building blocks at www.firebirdbio.com. So if you want to try any of these out yourself, it's available to you. And we've learned about genetic biopolymers in general, but we've also, for example, learned how to search for life that's alien, because we almost have created our own alien genetic system. For example, if we go looking for Spock or Sarek, not his mother, of course, she's from Earth, but the genetic biopolymer that they encounter will have a repeating charge in the backbone. It might be a positive charge or it might be a negative charge, like what you have in your DNA and myself as well. That's a universal feature for all informational biopolymers working in water. And we know that as a consequence of pursuing this grand challenge. Now, of course, the building blocks most certainly would not be the same as in Terran DNA. The building blocks, however, in any informational biopolymer must have the same size and shape so as to fit into Erwin Schrödinger's aperiodic crystal structure. More on that in a minute. And we know relatively how such systems could emerge in a rocky world with specific minerals around. And if you are interested in knowing more about this, we published a paper a couple years back on how to detect Darwinisms from molecules in the water that's coming out of Enceladus as plumes on Jupiter's moons and in other planetary water lagoons. So now that's great. We can make those molecules, we can make those and learn things, but there are actually 5 requirements for them to work for an alien life form. Those molecules must be able to store information with regular rules. By the way, that's helped by having this polyelectrolyte charged backbone. The information must be transferred to other function-capable systems. The Aegis expanded genetic alphabet must be able to deliver information to a receiving system that has a selectable phenotype. The biopolymer made of Aegis building blocks must be able to evolve. And of course, to complete the story, the most difficult thing is the system must be self-sustaining, able to find its own food. Now, we actually don't do self-sustaining in our laboratory, in part because our colleague Floyd Romersberg published a paper a number of years ago, and all he got was Andrew Pollock on the New York Times, sparking hope and fear. There's a layman's perspective on this is that somehow doing this is dangerous. So we don't do self-sustaining, but in this paper that we just published last year, where we had an 8-letter DNA and RNA system constructed, 8 letters is called Hachimoji, from the Japanese Hachi, which means 8 in Japanese, and moji means a letter in Japanese, like an emoji. And since the first author was Shuichi Hoshika on this, we call this 8-letter subset of Aegis Hachimoji. But it's 8 of the 12 letters that are possible if we were to use all of the hydrogen bonding patterns possible within the Watson-Crick pair. Now, I can't give a TED talk with too much data, but I'll put a little bit on because these are dots on lines. We can make lots and lots of duplexes which contain not just the AT base pair and the CG base pair, but also the BS base pair and the PZ base pair. Those extra base pairs are drawn right below in the pretty colors. And if you make enough measurements, you can develop empirical rules that will predict the stability of a duplex DNA. Predict its thermodynamic stability as in terms of free energy, which is the left-hand plot, or on the right-hand plot, it predicts the melting temperature of the duplex. On the horizontal axis, the vertical axis is the actual measured melting temperature. And you can see that the correlation is quite good. In fact, the Aegis pairing is as regular as AT and GC. In fact, AT, because it only has 2 hydrogen bonds joining the bases, is one of the outliers in this, not the funny bases that we've done by chemical synthesis. So that's great. We can store information on an 8-letter genetic alphabet with regular rules with the Aegis alphabet. The information must be transferable to other function-capable systems. That's our second criterion. That is, there's no use just storing the information. It must also move DNA, RNA, proteins. And we're able to transcribe this 8-letter DNA to make 8-letter RNA. Now, this is not trivial. It turns out that the natural enzymes that you get from 4 billion years of evolution on Earth with 4-letter DNA doesn't actually accept 8-letter DNA. But we can mutate the RNA polymerases to make them accept 8-letter RNA, make 8-letter RNA from 8-letter DNA. And then we can make all these triphosphates and we can develop the analytical chemistry. And what you're looking at here on the left is a bunch of radiochemical data proving that we have made the molecule on the right, which is actually that folded RNA molecule, which has 8 different building blocks in it. That's made by protein polymerases that we have engineered. So that's great. The system is able to store information with regular rules. Information must be transferable to other function-capable systems. The receiving system, however, must have a selectable phenotype. And what I just showed you, actually, that molecule, it does. It actually will bind specifically to a fluorescent molecule. Once it's bound, it glows green. You're looking on the left extreme and right extreme are 2 controls, but the middle 2, you're looking at the glowing of a functional RNA molecule built out of an 8-letter genetic alphabet, coming from the grand challenge effort to design an alternative expanded genetic system. Well, having shown that the receiving system is able to act as have a selectable phenotype, we must be able to say that the system evolves. So I mentioned already what was necessary, what we learned about the universal structures of evolvable systems. One is they have to have this repeating charge in their backbone, the so-called polyelectrolyte. The second criterion is from Erwin Schrödinger, that the building blocks of a evolvable genetic molecule must fit into an aperiodic crystal structure. And what that means is that replacing one pair by another as you change the information encoded in the system cannot distort the double helix. Well, collaboration with Millie Georgiadis at Indiana University, here are the double helices of 3 different self-complementary molecules built from 8 different nucleotides, aegis. And we can spend the next hour discussing it. These are within experimental error the same structure, replacing a ZP pair by a BS pair does not change the structure at all. In fact, the aperiodic crystal structure is violated the most by AT pairs, because that is the AT pair has only 2 hydrogen bonds, not the 3 hydrogen bonds that all the others have. So that's great, that's science, but correct theory also supports technology. And we can put the artificially expanded genetic information system to work. And let me just show you a couple of examples. So very often you want to do polymerase chain reactions in your lab, PCR, and often you want to do them multiplexed. But the problem is if you start putting, say, red primers and green primers and blue primers at high concentrations into a multiplex to amplify a red gene, a green gene, and a blue gene, sooner or later the primers go do mischief. They wander off and maybe bind with mismatches to a purple gene, and they give you junk. And this is, of course, a gel that everybody knows whenever you see amplification. So here is seeking to amplify 3 cancer genes. We have artifacts that are running slower than the bands, which are the ones coming from primer-primer interactions. We have artifacts coming from off-target primers as we increase the number of PCR cycles. Of course, this is a mess. Now, one way people try to manage this mess is by doing what is called nested or tagged PCR. What they do is they initiate the PCR with very small amounts of red, green, and blue primers, and but those contain tags. They're black tags. And so after the first round of PCR amplification occurs, the amplicons have these black tags on the outside of them. And those black tags are now used as primers to carry the PCR. Of course, the external black tags, the external primers, cannot bind or complement anything in the genome. And of course, that's hard to do because pretty much all 16 mers are present, say, in the human genome. But what do you do when you want orthogonality? Well, of course, you can put some of the components of the aegis system, some of the aegis nucleotides can be put in the external primers. And that's what we've done here to clean up this mess. And so now the external tags, of course, the PCR is initiated with standard molecules, but then after a couple of cycles, all of the amplicons have these external tags with PZ pairs. And now the PCR can be carried with PZ containing external primers, which at high concentrations, it makes no difference. They cannot prime on any orthogonal natural DNA. And what happens is instead of having the mess that you see on the left, you have the 3 genes amplified cleanly on the right. And so this is how we do PCR without having to worry about PCR mess. And all of this is available to you, as I mentioned, at www.firebirdbio.com. By the way, you can do this in infinitely large multiplexing. This is a case where we have published a paper to detect 21 mosquito-borne viruses. You can capture a mosquito, you can squish one mosquito here, and you can decide whether he detects or she carries Venezuelan equine encephalitis, dengue, any of a number of other things. It becomes very easy to multiplex PCR. And in fact, this aegis so-called orthogonality, that means that aegis oligonucleotides do not bind to anything standard, has enabled a $1 billion diagnostics project to measure the viral load of HIV, hepatitis B, hepatitis C in patient blood. And this was sold for many years by Siemens. It's cheaper to run than PCR. The problem that you have if you have the sandwich type of assay where one analytic DNA molecule or RNA molecule binds to a capture strand and holds a signaling unit was that you ended up having background noise where some of adventitious DNA is always capturing the molecules that you're trying to detect either or capture on a support. And though the branch DNA assay did not see fewer than about 300,000 HIV molecules per milliliter of blood above noise. Now, if you have 300,000 HIV virions per milliliter of your blood, you are a very sick puppy indeed. So how do you solve this problem? Well, we put Aegis components into DNA parts that assemble the signaling nanostructure. And the Aegis orthogonality separates the analyte binding from the concentration in signaling. And so you can drop the signal level down to about 8 molecules per milliliter of DNA. About 400,000 patients per year had improved care, about $1 billion in lifetime sales. And by the way, this is now in multiplex assays for those of you who are interested to detect the SARS coronavirus, the very virus that is preventing us our meeting face to face. And this over here is the SB base pair that was used to clean this assay up. But I promise you synthetic Darwinism. Remember, replication with errors, where the errors are themselves replicable. People do this or have tried this with 4-letter DNA and RNA, but the results are disappointing. These are people like Andy Ellington and Jerry Joyce and Jack Szostak, the Nobel Prize winner Larry Gold, and others. And the reason for that, again, is because DNA has little of the functional groups and information density of proteins. So what do? If you want DNA with higher, more functionality and more information density, well, of course, different question, same answer. All you have to do is put Aegis Darwinism into this. And this is just a schematic done by Liqin Zhang and Kwame Sefa and Zuni Yang and Wei-Hung Tan to select for molecules that bind specifically to liver cancer, in this case, also breast cancer, various toxins. We've applied this in many cases. And what you do is you make a library, about 10 to the 14th different molecules. Most of them do not bind to the cancer cells. But you throw the target cells in and you go and throw away all those that don't bind, you recover those that do, you PCR amplify them, you might do a counter selection. But then you repeat this cycle 10, 12 times, and eventually you perform 6-letter, 8-letter deep sequencing on these to discover within the 10 to the 14th different library components which ones bind to the cancer cells. And you can get numbers. Here's a whole bunch of these sequences that do, with various dissociations. One thing you should notice here is that I've ranked these in terms of weaker and weaker binding going down the chart. When you get to the weakest bindings, there is no PZ, which is the red letters. When there's the stronger binding, there is PZ. The best binders contain these. So the nitro group, the more nitro groups there are, the greater the specificity. This is something meaning that the course design plus the redesign of our, based on our better understanding, our redesign to get better nucleic acids has created a new chemical system that does what we value in biology, the ability to be replicated with errors, where the errors are themselves replicable, and get evolution and adaptation, science and technology. And of course, now Aegis DNA is today being used or evolved to bind and cleave, actually now, coronavirus spike proteins. This is our famous SARS-CoV-19 structure. On the right, you are seeing the binding of an Aegis enzyme, which is in a spray bottle, not caustic, can contact the skin, the eyes. This is not Clorox or bleach. We're not asking you to take that internally, but you could drink this stuff. It's really not toxic, but it does inactivate the virus. It also tags it with a fluorescent marker. So that's what you've seen today, an evolvable biopolymer. It's combining phenotype with genotype to generate both science and technology. This grand challenge synthesis drives discovery and paradigm shifts. Universal rules come out of this grand challenge effort to understand genetic biopolymers universally. They're used to support the search for alien life. It gives ultra clean PCR, detection of viruses in mosquitoes and blood. It evolves to give molecules that selectively bind breast, liver, other kinds of cancer cells. And it evolves to give catalysts that destroy other biomolecules, including viral biomolecules. Now, all of this is available to you. So that's all I have to say. Thank you for listening. We're trying to understand the thing called life. This is what we do in my laboratory. On the right-hand side is the synthetic biology that I've already described to you. Of course, we do use this going out to try to explore the solar system to find aliens. But synthetic biology, artificial genetic systems, science, and technology. And thank you for your attention.